Half a day. I'm Rebecca Hamner, the Conservation Genetics Program Manager for the CNMI Division of Fish and Wildlife, and I'm based on the beautiful island of Rota. When I joined DFW 2 years ago, there were a few genetic collaborations primarily focused on taxonomic clarifications. Since then, I have identified many more areas where genetic tools can fill in additional knowledge gaps or aid with management actions. My first task was to review all 60 species of greatest conservation need that were identified in the CNMI Wildlife Action Plan, and develop a strategic plan to efficiently address conservation genetics needs over the next 10 years. And since that was completed in 2019, I have been implementing our priority projects. Those 60 SGCNs include several species, subspecies, and distinct population segments, as well as species, and the broad categories of food fish and all staghorn corals. 33 of these were terrestrial species and 27 are marine, with just a handful of them pictured here. For each of these species, I evaluated what conservation and management questions had been identified that genetic tools could answer. So what information can genetic tools provide? One of the most basic questions is, what is it? DNA barcoding can be used to identify both native and non-native species, similar to how barcodes are used in the grocery store. For example, if you scan this top barcode at the register, you'll get Rhoda's favorite coffee to help you wake up this morning. Or for those of you at the other end of your day, maybe you'd rather pick up this item and crack open a can of Rhoda's favorite beer. Well, DNA can be used in a similar way, using ATs, Cs, and Gs instead of bars and numbers. So if you come across this sequence, you found Rhoda's endemic partulid snail, whereas a different sequence might indicate a brown tree snake, which we hope to never find in the CNMI. By comparing DNA sequences among areas, we can also determine if our critter of interest is unique to the Mariana Islands or even to particular islands within the archipelago. We can also use DNA to detect where a species occurs across the land or sea and map its distribution. We can determine if groups of individuals are connected or isolated from each other. We can determine if there are genetic threats to a population or species, such as a lack of genetic diversity or inbreeding of closely related individuals. And we can also use DNA from the feces of animals to determine what they are eating. While there are many more things we can learn using genetic tools, I'll stop here because these cover the main questions of interest that came up during my review. So getting back to that review, what critical questions for our 60 SGCNs can be answered using genetic tools? 15 species need taxonomic clarifications, 6 have unclear distributions, 51 have questions about connectivity among islands or to the greater Pacific region, 37 need to have genetic risks evaluated or managed, and this includes 16 that have small isolated populations, often on a single island, 15 species that have been translocated in the past or will be in the near future, and 6 captive populations for which breeding among close relatives can become a problem. Beyond species-specific needs, I also identified the need for a reference database of DNA barcodes for local species. Many of our species have never been sequenced at all. This database will aid with larger scale biodiversity surveys that can use DNA shed into the environment and collected from water, soil, or even air, which will be especially useful in the Northern Islands where we don't get very often, and when we do, it's only for very short periods of time with lots of objectives to accomplish. I also have a feeling that as we start filling in more barcodes, we're going to start identifying a lot of new endemic and undescribed species. When we know what native biodiversity we have, it will help us to identify non-native species that might pose threats. These non-native species can also be detected by environmental DNA surveys, perhaps even before they are visually detected, and environmental DNA surveys might also aid with monitoring their spread. As you can see, there is no shortage of projects that genetic tools can aid with, but we have limited funding and people power, so where do we start? To prioritize these projects, I considered the biological factors of endemism, distribution, and abundance, as well as management-related factors, including management utility, or how would learning this information actually change what we're doing to conserve the species, the number of questions that could be answered by the collection of genetic samples, the ease of sampling, if we already had an established collaborator to facilitate project completion, and the cost of the project. Priorities were designated as projects that met one or more of the following: ongoing projects with established collaborators. Let's get those done and dusted before we start new projects. A genetics need score of 14 or higher. And this is just the numerical translation of the biological and management considerations that I mentioned before. And combination projects that share field effort and/or answer questions about multiple species of greatest conservation need. Using these criteria, 27 projects were designated as priorities for 2020 to 2030. This may sound like a lot, but we have many collaborators who are leading or contributing to most of these projects. 20 of the projects were terrestrial, and I'm excited to say the first one was done and dusted just recently, with Cisco and Hadfield publishing the description of Partula ludaensis, the snail pictured in the upper right, which you might have heard Dave Cisco present about last week. A second project is almost complete, with our collaborators at the Smithsonian Institute finalizing a manuscript assessing the taxonomy and inbreeding of the Saipan reed warbler. 14 more projects are ongoing, and 4 are waiting to be started. On the marine side, we had 6 priority projects, one of which is also nearly complete, with a manuscript assessing the taxonomy and endemism of 3 turbo snail species, which is being drafted by Gustav Pauly at the University of Florida. There are 3 additional projects that are ongoing, and 2 are yet to be started. And our final priority, the genetic archive and reference database for local species, has been established and is very slowly building up data. Now, I'll just briefly highlight a few of the ongoing projects that DFW has active objectives related to at the moment. This includes clarifying taxonomy for Aliling pulan, the turbo snails, Igigi, the Micronesian honeyeater, Naabak, the rufous fantail, Akalea, the partulid snails, and Achiak amoya skinks. Aside from the nearly complete turbo snail project, all others are in the sample collection phase. Now, the biggest project I'm getting going at the moment is to determine the distribution and ecological associations of the Rhoda blue damselfly, Dulalas luta. Very little is known about this federally endangered species endemic to Rhoda. So we're going to start by determining the extent of where it's found. To do this, I'm using environmental DNA that has been shed into freshwater habitats upon which this species relies for breeding. As you can see on the map in the upper right, there are only 2 areas with freshwater on Rhoda, the streams of Talakaza and a few man-made ponds at the Rhoda Resort. This species has never been sequenced before, so I'll start by non-lethally sampling a few voucher specimens and establishing its DNA barcode. Then we can go out and collect water samples and see which ones contain DNA that matches the Rhoda blue damselfly's barcode. The idea being, water samples taken from areas where the damselfly lives will contain its DNA. In addition to determining where the Rhoda blue damselfly is, we're also asking, why is it there? To answer this question, we will collect data on habitat associations, including water characteristics, canopy cover, and vegetation types, as well as assessing community composition to identify potential competitors, predators, and prey. For this objective, instead of looking for the known sequence of a particular species, we will use DNA metabarcoding to ask, what are all the species that are here? This metabarcoding technique will also be used in a slightly different way to determine the diet of the Phenihimganas, the Pacific sheath-tailed bat, and the Chachaguac Mariana swiftlet. This is part of a project led by Leroy Sablon to ensure that there is ample prey for these 2 species as plans to reestablish them to their historical range on Rhoda are being developed. The idea here is that the guano of these species will contain DNA from the prey that they consumed, and we can sequence that to learn what they are eating. Even these 2 metabarcoding projects that I just described highlight the importance of having a reference database of local species barcodes. Some of our species are quite well known and studied, while others are not. This database is something that I am building to aid with DFW's projects, so we don't get back a bunch of sequences and go, well, it's a beetle, but I'm not sure which one, but rather that we can put an actual species name to as many things as possible. And I think this will be useful to others working in the Marianas as well. So I encourage anyone who's working on barcoding or metabarcoding local species to get in touch with me. I've communicated with a few of you already, and I'm planning to organize a meeting to discuss ways we can build on each other's barcoding efforts and all have access to the most up-to-date reference database to help ID species for our various objectives. And with that, I'd like to say Suzuos Masi. Thanks to the U.S. Fish and Wildlife Service State Wildlife Grant Program for funding the creation of the CNMI Conservation Genetics Strategic Action Plan. And additionally to the competitive SWIG and WISFR programs for funding our ongoing projects. Thanks to all of my coworkers, collaborators, and community members who make this work possible. And if you want more details, you can search for the CNMI Conservation Genetics Strategic Action Plan. My email address is also in the lower right if you'd like to contact me with any questions, collaboration ideas, and I'm happy to answer a few questions now live.